great pleasure to be here to finish off today. We've had some great talks, and it's my job to sort of bring things to a conclusion for today, barring a discussion and a photo, by giving you some ideas about how ICDA might approach that final mile of taking all this wonderful information that comes from genetics and genomics and think about ways in which you might scale it for application to patients. As Cecilia said, I recently moved to pharma 3 months ago, so I wouldn't be presumptive enough to speak on behalf of all of pharma based on about 105 days of expertise and experience, but this is really a synthesis of a lot of work that's come from others that named on this slide and beyond, who've participated in a number of discussions over the evolution of ICDA and on telephone conferences and so on. So the challenge is the one that's exemplified on this slide. It's to take two broad types of large-scale data that are already in existence and which we, as we've discussed today, will improve in both scale and scope over the next few years. The data on genotype-phenotype associations, which essentially takes alleles and relates them to phenotypes across exposures, and genomic and functional data, which takes alleles and relates them to function across cell types and cell contexts and so on. And the synthesis of all those, we hope, will lead us to an improved understanding of how diseases come about and how we can use that information for clinical benefit. And in terms of translation, it's pretty obvious to see that what we're driving at here is getting towards a comprehensive perspective on the mechanisms that underlie medically relevant phenotypes, using that information to identify novel therapeutic targets and aid their development into safe and effective medicines. It's also, and we haven't talked about this too much, and I didn't want to neglect this, although I'm not going to talk too much about it otherwise, is of course using genetics and these other tools to characterize the modifiable risk factors that are causal for disease and might be amenable to both population-wide and targeted interventions. The challenge, of course, is that whilst the two on the left are already well scaled up and are ripe for further scale up, when we start to think about how we use that information in the context of disease and disease mechanisms, we're in that kind of final mile analogy where the work that we do around disease mechanisms and developing new medicines has historically been and continues to be a largely bespoke exercise, and one that looks from the outside pretty resistant to scale up, and I'll come back to that in a minute. So the challenge, I think, within ICDA in this particular area is to identify those aspects of translational research that can be moved upstream in terms of scalable data generation and data sharing. It's a bit like if we were 10 years ago and you found a GWAS hit, you then had to indulge in hand-to-hand combat on the variant that you thought was the lead variant to get all the functional data on that variant. Now, of course, you can look up a good deal of it and make mechanistic hypotheses already based on the data that's out there in the public domain. So to what extent can we do the same sorts of things around disease mechanisms and drug development? So the way that we broke it down is summarized on this slide to divide it into some things we thought were quite proximal and mechanistic. I'll tell you what I mean by that in a minute, and others that were more distal and translational, and again, I'll come back to those. So in terms of the first group there, the ones on the left there that I've labeled mechanistic, we think there's an important place for ICDA and for broader collaborations in helping to articulate that dialogue, that iterative dialogue that needs to happen between disease researchers who may be invested in particular disease areas and particular mechanisms, and the very broad generation of large-scale data that comes out of the things on the left there. I'll just give you sort of 4 examples of, so you can see what I'm driving at here. The first one is to make sure, for example, that large-scale genetic data generation is keyed into the translational phenotypes that matter, particularly for pharma, of course. Those have been historically difficult to ascertain, have not necessarily been the substrate for most of the genetics that's happened so far, as we heard from Matt. Those really represent the areas in which we really need to identify the genetics of progression, for example, genetics of drug response to aid what we're trying to do. I think it's also important that large-scale genetic data is generated on the cell types and the states and the contexts that are relevant to disease-specific investigators. It's not automatically giving the truth that large-scale data generation is necessarily going to go to exactly the kinds of cell types and the contexts in terms of exposures that turn out to be most germane to diseases. Of course, there's an iterative aspect in here that the data generated will help us key into what those might be and call out the need for further large-scale data generation. We need to make sure that there's interpretation of large-scale functional data in the light of what we know about disease biology, so that for a given GWAS locus, we're looking at the right cell type in the right context for functional interpretation. And I think another area is in terms of all this large-scale data generation, which is designed to inform on mechanism of disease. We need to have truth sets against which we can judge the performance of those different methods, so that there's a joint effort here around data comparison, integration, calibration, developing metrics of performance, driving machine learning, and so on, by having rounds of data generation and validation in disease-relevant data sets. And just to amplify a couple of comments, I've worked historically on type 2 diabetes. We've known from physiology for quite some time there are multiple processes involved in risk of type 2 diabetes, and we have genetic substantiation of pretty much all of them from one or other genetic locus that's been teased apart, and we know the mechanisms through which it operates. So that when we run, certainly in bulk, data sets and find enrichment for islet enhancers across GWAS, type 2 diabetes GWAS, that tells us, for example, that islets are important, but shouldn't lull us into belief that all of our loci work through islets, because clearly if we follow that path, we're going to make some wrong functional inference. So we do need to tease apart for each locus at every disease that we care about what the mechanisms are and the cell types that are involved. And then just to amplify the last point I made on the slide just before this, we, for example, have multiple ways of getting from a variant to gene. We've heard about some of these today. I've listed some here. We've heard about some others that I haven't listed here. We all know that these are powerful techniques, but we all know that they have incomplete specificity and sensitivity, and as we pile up more and more data to address those links between variants and genes, we aren't always going to end up with results that are entirely coherent and consistent. So we need to do a job, ideally around identifying truth sets that allows us to get to understanding which of bits of these information, which of these approaches are most valid, useful, coherent, how we can combine them, and how we can use them to get more robust inference about the effector genes that lie downstream of regulatory GWAS signals, for example. So that's the bit on the left. If you turn to the bit on the right, we identified 6 main areas where we would want to see development, or 6 types of data we really need to see, and those are shown in brown on the right. The ones in the middle I've grouped together because those really do flow from having a better mechanistic understanding of what's going on at a given signal. The pleiotropian stratification angles I'm not going to say too much about, because those largely flow from the genetics alone in ways that we've heard about already. And of course, the reason why those 6 are important, particularly from a pharma perspective now, is that they each play into important components of the drug development process, not just target identification, prioritization, but all sorts of other bits of information and tools and resources and markers and assays that we need to get a target through to a medicine. This audience will be familiar with the need to do better, that pharma is afflicted by both an attrition problem with too many drugs that fail during clinical development, which obviously adds a huge burden to the cost of successful launches, and also by an innovation problem, that more and more of the medicines are just tackling the same old pathways and often fail to differentiate from the standard of care. And also, you'll all be familiar with the notion that genetics should be a tool for helping here. We saw the Nelson paper presented earlier today. There's a more recent update and reanalysis of some similar data in bioRxiv that reached similar conclusions. And we also have many and a growing number of good examples of genetic discoveries that have keyed into new therapeutics. So in terms of thinking ahead and thinking about the things that pharma would like to see, and hopefully disease researchers would like to see that could enable those efforts going forward, we identified 5. The top 2 are ones that we've discussed extensively today, so I'm going to concentrate on the bottom 3, which is around detailed mechanistic studies, around functional assays and biomarkers, and around data integration for genetic discovery. And because Robert Plenge likes acronyms, I call this the 3M problem, and I've labeled what those 3M's stand for in yellow there. So the first is around deep dive into mechanism. We heard from Ramnik, we heard from others about the approaches that you are taking, and to simplify the kinds of approaches that many of us are taking and making it probably simplistically linear, but these are the kinds of steps that many of us are going through in trying to translate genetic signals for a disease, a GWAS for a disease, into pathogenetic mechanisms, understanding the phenotypic spread through PheWAS, trying to get to the cell type and state and regulatory effect, trying to find the downstream genes, establishing in some sort of perturbation assay that there's a causal connection between those candidate effectors and the disease we care about, and then trying to knit this all together into pathogenetic mechanisms. And I think what is obvious is that although the steps to the left there are fairly generic and you can do the same sort of things across disease areas, the further you step to the right here, the more bespoke it is. And just to give you an example, some of the work that we've been doing in type 2 diabetes with Anna Gloin and others around the planet, I won't go into the detail here, but it's pretty obvious that what we've invested a huge amount of time into is taking the tissues that we think are relevant for type 2 diabetes, having to generate the GTEx and ENCODE and epigenome roadmap for pancreatic islets, because those weren't included in those community projects, looking at differentiation of iPSCs to understand the regulation in those setups, building cellular models that we believe in, building animal models we believe in, establishing phenotypic readouts that we think have some relevance to disease. And anybody working on any complex disease will have their own kind of set of things that they want to do in this space, and their own set of models and assays and readouts that they need to develop, which does make this look difficult to scale. Nonetheless, I think there are a number of areas where we should be thinking about opportunities for scale-up and sharing. Commonalities in analytical strategies around network biology, machine learning, for example. Common frameworks for mapping disease biology. This is really picking up on something that Matt says around the idea of the omnigenic model, for example. Whatever you think of that, it's at least an attempt to frame everything we do within some sort of common framework. Probably not the end story there, but an opportunity to think about ways in which we can map different diseases into similar frameworks. Community standards, metrics of success and validation, development of more sophisticated cellular and tissue models, and exploring of shared biology, because of course, as we've heard, there are a lot of fundamental processes here that map across different cognate areas and could be opportunities for shared activity. Which diseases should we focus on? We struggled, we've talked about this endlessly. I think the important thing to recognize is that there are some diseases that are further down the path towards a fully genetic description and use towards medicines, but those are often the ones which there are already many existing medicines. So whilst we should clearly take diseases like diabetes and IBD forward with gusto because of what they will tell us, we mustn't neglect other diseases, the fibrotic diseases, for example, where there's massive clinical unmet need and where we have very few tools at the moment to take those forward, but that's really where the opportunity may lie for the biggest advances. It's pretty obvious to say that we need better assays and markers right the way through the drug development protocol. Jay talked about some of the ways in which we can address variant level, scale up variant level analyses through MAVES and so on, so that we get better understanding of the relationship between variant and function at the gene level. We need to then get into gene level, genetic and chemical screens, which involves developing better cellular and in vivo systems and richer molecular and cellular readouts. And then, particularly as we start to have a target that we want to take forward, we need a range of clinical biomarkers. In many companies, it's the case that without a translational biomarker for which you can measure target engagement, that the trials won't go forward. So we need to access those biomarkers. We have plenty of opportunities around PQTL data sets, extending PheWAS more rigorously around toxic biomarkers, using population risk scores, polygenic risk scores, I should say, in populations and cell systems to enumerate these. And then the last area, which is dear to Robert Plenge's heart, and you'll probably have seen him write about this and blog about this, is understanding the magnitudes of effect. We're quite used to saying for quite some time that you can't say very much about the magnitude of drug effect you might expect from a GWAS locus, because they're all small and the effects of a drug might be that much larger. As we get more and more data on rare variants of larger effect, as we establish phenotypes in human knockout models, of course, we do have better and better tools for being able to calibrate the relationship between the genetic effects we see and the potential therapeutic effects we see, with some assumptions, of course, that there's an equivalence between those two types of perturbation. And so I think there's a definite need to bring together multiple types of data on the relationship between genetic perturbations and phenotypic outcomes across systems and across readouts, and to have that in one place. To some extent, that's something that Open Targets is doing quite well. We've had some discussion within the group as to whether we need to do something different and additional to that, but that's certainly a subject for further discussion. So let me finish there and really just pay testament to those who've helped me think through some of these issues. As I said, I'm new to pharma, and Sally and Robert and many others have contributed to this. I'll just leave up perhaps this slide, which summarizes some of the areas that we thought would be priorities for translational research through ICDA. Thanks.